All right, good afternoon, everybody.
Good afternoon.
Ready for the last day? Where's our guy? Dan Simon, how we doing? All right, well, definitely study hard. This exam is obviously kind of a different, it's kind of market runs and also to reiterate that I tend to test on like function in general, right? But when you don't get too bogged down in like the exact origin and description of every single little muscle, right? You're gonna know one thing, no reaction of the muscle, okay? And then kind of as far as like where the muscle comes from, as far as which bone and which aspect of that bone, and then where it goes to, which aspect of that bone. That's really what I like to focus on. I don't really have any particular questions on like the exact bone of dominance or muscles that we're going to insert on, but it's really thinking more about the action, about the part of the bone and the symptom, so the direction across one of the joints to produce that action. Definitely focusing on that for the moment. And then the diagrams, well, knowing being able to identify them visually. Any other questions on that stuff? All right. So we're gonna dive into this stuff here, which is obviously the stuff to start off exam 5, okay, for our next exam, so not 4 exam on Friday, but this will kind of backtrack us a little bit from where we started. We're talking about muscle contraction. We talked about excitation, contraction coupling, and then top group section, all of that. This is kind of one step before that, right? All the stuff that happens at the junction between the nervous system and the muscle, so the neuromuscular junction. Okay, so when we look at the neuromuscular junction, it really contains 3 different components to it. Okay, because it's called the synaptic terminal, which is where the ends of the neuron, okay, and that axon, the end of the motor neuron axon. And this has a bunch of vesicles there, which is just like little miniature cells, right? Like little double membrane cells that contain a bunch of acetylcholine. Acetylcholine is the neurotransmitter that motor neuron into the space between the muscle cells. That is muscle cells. Acetylcholine is definitely one of the most common neurotransmitters throughout the whole body. You see a lot of the brain as well. We'll talk more about other neurotransmitters at the end of the semester. But yeah, so that's the one side. That's the nerve side of the neuromuscular junction. And then we have this space between the 2 cells. Right? That's the synaptic cleft, the fluid-filled space between the muscle cell and nerve. Okay, so this is that space for diffusion of acetylcholine across that space. And this contains an enzyme called acetylcholine esterase. Okay, and its job is to break down acetylcholine. And so after it's been okay, and so this is what's going to help with relaxation of muscle. Okay, so we got the nerve part of the neuromuscular junction, the space in between the 2, and then the muscle side is called motor input. Okay, and this is that just part of the muscle cell membrane, part of the sarcolemma, heavily folded. It gives us more surface area. And the reason we want more surface area is so we can put more proteins on that surface area. And the proteins that we're talking about are acetylcholine receptors. Actually, the acetylcholine receptors embedded in the membrane, right? So that's what that acetylcholine is going on. These acetylcholine receptors are also ion channels. Okay, so when acetylcholine binds to them, that changes them in such a way that it opens up the channel that is a part of that same receptor, and then allows ions to run into that channel. And we'll talk more about all those details here in a second. Okay, so go ahead and kind of label the different things we just talked about there. and then you can answer this question here. And it's one of the letters, not one of the numbers. Okay, so the numbers are kind of like the three general regions, and then letters are other specific things found there. Okay, so you're sending in the answer to that question there in the top. All right, go ahead and send that in on the All right, I think there's probably still another 8 of you or so that haven't sent it in yet. Go ahead and send something in. All right, looks like that might be most of you. Leave it open for a sec, just in case. So most of you are saying C. Yeah, so C, and what do we call that? Huh? Receptor, yeah, there we go. Acetylcholine receptor, for sure, okay? All right, so we'll get to that one I'm covering here in a sec. Let's start with the numbers first. So what do we call number 1? Pre synaptic. Okay, and then we got number 2. What do we call that one? Flat, which is really just the space between the two. All right, and then what do we call the muscle side of that? Number 3. Okay, so we got those 3 regions, and then let's start with the other little parts there. So A, these are those vesicles full of acetylcholine. Okay, any ideas what B is? Now, this is just a depiction of that enzyme that helps us break down acetylcholine. So acetylcholine, that's great. Okay, you guys already said what C was. That was that's the acetylcholine receptor, which also functions as an ion channel, right? So neurotransmitter gated channel is just kind of a general way to describe that, which would be an ion channel that is gated by a neurotransmitter binding to it or not, right? So that's what opens or closes the gate, is whether or not that neurotransmitter is bound to it. Okay, and then D there is something we haven't talked about yet, but this is a voltage gated ion channel, sodium channel. And we'll talk more about those coming up next week. But yeah, so we have different ion channels on the regular part of the muscle cell membrane. versus the motor itself. Okay, and then E, we have a different kind of voltage gated channel, it's a calcium channel, voltage gated calcium channel. Talk about that one in a second too, when it comes to the active. So when this active potential travels down the neuron, it's gonna change the voltage around these calcium channels. And that is the trigger to open up the calcium channel and do that voltage. Okay, and we'll talk about the role of calcium here in a second. Okay, and then lastly, F, what's that? Pseudocolon, right? Right, right. So that is our neurotransmitter, pseudocolon. Okay. Questions on that? Got it? Okay, so that kind of sets this up. That's the kind of structural stuff we have going on in the neuromuscular junction. And before we get into actually what's happening in the neuromuscular junction, we need to kind of talk about this idea of electrical excitability in cells and what's known as resting membrane potential. Okay, so this little image here is showing the cell membrane right here. Okay, intracellular on this side, so that right side means inside the cell. The left side is outside of the cell, right? We see a membrane there in the middle. Okay. One thing that's really important to understand is what we kind of have a lot of on the inside of our cells, what we have a lot of on the outside of our cell. Okay. So just by looking at that, and it says there on the left, you can tell that we have a lot of potassium on the inside of our cells, right? So K plus there, right? So inside is high potassium. Okay. But there's not much sodium. There's one sodium there, not much, right? So low sodium, high potassium on the inside of our cells. Okay. But then we also have these ones here. Those are the negatively charged proteins. Okay. So inside the cell, we got lots of potassium, we got a little bit of sodium, and we got these big negatively charged proteins. Okay. On the outside of the cell, kind of the opposite as far as sodium and potassium goes. We got lots of sodium on the outside of our cells. Okay. Some chloride, but we don't really need to worry about that so much. But that's the big deal. So low potassium, high sodium. Okay. The reason this stuff is so important is because the way that we get electrical signals traveling down cells is all about ions passing from one side to the other and changing the voltage. Okay. So when we have a what's known as resting membrane potential, it's just kind of what our nerve cells look like, what our muscle cells look like when they're at rest. We have a negative charge on the inside of the cell, positive charge on the outside. Okay, so that's a really important thing to remember. Again, so negative over here on the inside, positive on the outside. Okay. Now we'll talk more about this, but there's a couple primary reasons for that. One of the reasons why it's mostly negative on the inside of the cell, one of those reasons is these guys right here, these negatively charged proteins. Okay, so that's kind of reason number one. We have these big negatively charged proteins that are stuck inside of our cells. Those can't move across the membranes, they're too big to do that, so they're just kind of stuck in there. Okay, so that's reason number one. Okay. If we go here, reason number two is shown here. And so, yeah, let's go back actually. Reason number two is actually shown here. That next slide will be reason number three. But reason number two is that there's special channels that allow potassium to constantly leak out of our cells. Okay, we have both potassium and sodium leak channels in our cells, but we have more leak channels for potassium. Okay, and so when it comes to ions moving in terms of diffusion, they're going to move from a high concentration to low concentration, right? So we have the ability for potassium to move. Which way is it going to move? We just open up a channel for potassium to move. Which way is it going to move? In or out of the cell? Out, absolutely, right? It's going to move out. Okay. And so since we have some positive charge moving out, but we don't have as much of a positive charge moving in because the sodium doesn't have a leak channel, that results in a net negative on the inside of the cell. Then we're losing more of the positive charge than we are gaining, right? So that is reason number two for why the inside of the cell is negative, right? Number one was the proteins that are negative in there. Number two is we have more leakage of potassium, which means it leaks out. So we're losing more of the negative charge than we are gaining. Losing more of a positive charge than we are gaining a positive charge. Right. And then reason number three is shown on this slide here. This thing here is called the sodium potassium pump. okay, also known as sodium potassium ATP. Okay, we have these little proteins in all of our electrically excited cells, we have lots of them, okay? So they use an ATP and they pump sodium out and potassium in. Okay, where was there already more sodium, inside or outside? There was already more outside, right? Where was there already more potassium? Inside. So both those are going against the gradient, right? So what kind of transport is that called? Active transport, okay, and we got two options of active, right? We got primary active, secondary active. Which side do you think? There's another difference. Primary uses what? ATP, and secondary uses the concentration gradient of something else. Right, so in this case, we're using ATP, so this is a form of primary active transport. Okay. And so it pumps both sodium and potassium, but it does it in an uneven fashion, right? Because we got 3 sodiums going out and only 2 potassiums going in. So that also results in a net -1 to the inside of the cell every time it pumps, right? Because we're losing 3 positively charged sodium and we're gaining 2 positively charged potassium. So the net result of that is another -1 to the inside of the cell. All right. So again, those 3 reasons were, number 1, more leakage of potassium, number 2, those negatively charged proteins, and number 3, the uneven nature of these sodium potassium pumps. Okay, let me just show you this little video on this. It kind of walks us through some of the stuff I just talked about. That this will help solidify that. There we go. Okay.
The lung contains about 100 billion nerve cells called neurons. A neuron, like every other cell, has positively and negatively charged ions inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on 2 factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at -90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to -70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
Okay. So, um, yeah, so, let's talk about that just a little bit more here. So we've got, all right. Let me try this again. There we go. Okay, yeah, so that one didn't say the third reason, but it said the two reasons, right? It didn't talk about the proteins, but it talked about the other two, okay? So normal resting membrane potential is somewhere around -70 millivolts, okay? Let's just do this real quick to make sure everybody's paying attention. Answer that question there, okay? Which side's negative, inside or outside? All right, send that in. Okay, so 95% of us are saying, hey, the inside's negative. That's true, okay? For those three reasons we just talked about. So go ahead and talk about this one as well with the people on your team. And you can send in more than one answer. And yeah, talk about that and send in your answers when you're ready. There's a set of people on the team. If you're on the same page, everything we're doing. All right, so putting all your answers in, then go ahead and hit the send button to make sure. Looks like quite a few people still need to do that. All right, think about 10 more people still need to send that in. Just make sure you hit the send button. All right, good. Got most of the people there. So about 90% of you are saying A and C, and only about 40% of you are saying E. So let's talk about A and C first. So the fact that there's more passive leakage of potassium than there is of sodium, that one's definitely true. Okay, but that one has to be coupled with E, right? Because the fact that sodium leaks in and potassium leaks out, right? Because that means we are losing more of a positive charge than we are gaining, right? We put A and E together, that kind of tells us that. And then C, the fact that the sodium potassium pump moves 3 sodiums out and 2 potassiums in, making it a net negative effect every time it pumps, right? So those 3 factors are the 3 key factors that dictate resting membrane potential. Okay. All right. Questions on any of that so far? Okay, so we'll get into more of this later. And sorry, this is from, oops. I skipped that. This is from our old textbook. I forgot to cut that out of the notes this time, but you don't have that anymore. But I wrote there in we'll get there. But what we're talking about now is resting membrane potential. But in order to have an actual signal being sent down a neuron or down a muscle cell, we get what's called action potential. Okay, so this is very rapid, reversible, spreadable change in membrane potential in positive direction. So we started at -70, right? And action potential goes up in the positive direction. Okay, we can kind of see that here. We're down here, the rest of the membrane potential, and it kind of peaks up here at around positive 30. Right? And we'll look at all those steps later on. I think it's sometime next week we'll get into that. But this is really what's known as kind of a language of the nervous system. I think how like neurons interact with each other, how they interact with muscle cells, like gland cells, any cells that our neurons control. To do that via action. Okay, so we'll get into more of that, what that means later. But before we do that, we're going to talk about the events here at the neuromuscular junction. Okay, and So yeah, again, these are the steps that we're gonna walk through here. These steps are shown on these next slides. Okay, and what I'll do is I'll show this video here. Sorry, those little question marks are little arrows. Those get messed up sometimes. I'll show this video here that kind of walks us through these steps. And then I want you guys to use these steps on the slide along with the next 4 slides to kind of fill in what's happening on each of those. And then we'll go from there. Okay, so let me see if I can click on this one without getting kicked off the Apple TV. Not gonna work for us.
Or a bit of, well, I always want to find ways to save you money. So it lets me barely paid spokesperson.
There we go. Okay,
Nerve impulses.
do you want me to turn the light off? Can you see it? Is it better with the light off? How much you?
Also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
All right, so that kind of walks us through the steps there. So take a couple minutes here with the people in this room, go through those slides, kind of fill in what's happening in each step. And we'll come back together here in a second. Yeah, and you can actually answer this question at the end here after those diagrams while you're doing this as well.
Oh, right, oh, this is the channel.
All right, looks like most of you are writing in what's happening in towards the end. So let's go ahead and talk through this. I haven't closed the quicker question yet. We can get to that in a second. But let's talk through step one here. So these little pink lines, what do they represent? What is it? Action potential, right? Yeah, so we got action potential. Coming down the motor neuron. Right, that's kind of what's happening there. Right, when it comes down, it then interacts with these voltage gated calcium channels. Okay, so action potential opens. Voltage gated calcium channel. Okay, and then which way does calcium go? Into which stuff? Perfect. Calcium. Into the neuron. Okay, and the body actually utilizes calcium for many, many different cellular mechanisms like this. Just one of many, but calcium is the primary signaling ion that activates things happening inside the cell. So this is just kind of one example of that. That calcium goes in, it binds a special protein. They're going to make this vesicle move towards the end of the neuron. So these vesicles go down to the neuron, and we just fuse with the neuron, and then we dump out all this stuff right there in the synaptic cleft. Right. So Yeah, so we see that, so then, yeah, I guess we could also add in that, acetylcholine is happening in too, right? The calcium, recommend possibly acetylcholine, to be released. Okay, and then we see acetylcholine going across the membrane, or across the synaptic cleft, right? So acetylcholine then binds to the acetylcholine receptors. Okay, the video called those receptors ligand-gated ion channels. Okay, so we can gauge ion channels a whole bunch of different ways. We can gauge them by voltage, right? So voltage-gated ion channels. We can date them with a ligand. A ligand is just something that binds to it, right? So in this case, it's the acetylcholine. Acetylcholine binds to it, opens it up, and then it allows the ions to flow, right? And this one's kind of interesting. It's not specific to sodium or potassium. It allows both of them to go through. That's what we call a cation channel. It lets these both positively charged ions go through. But there's a much stronger gradient for sodium to go in than there is potassium to go out. Any thoughts on why that is? It looks like there's a lot of potassium on the inside, a lot of sodium on the outside. What would make it so there's such a greater rush of sodium going in when they both have the opportunity to move? Talk about that with each other for a second. What are your ideas then? Okay, who's got an answer for me? I'll see some hands raised. Who's got one? There you go. Okay, but they both are, right? Passing is going out, so they're both going with the concentrated gradient. What's another idea? Not related to osmolarity. You want that idea? There it is, absolutely. Right? We got two factors that drive ions. One is concentration gradient, that's just how much of each one we have. The other one is electrical gradient, right? So since the inside's negative, that's a positively charged ion, opposite attract, right? So we have the electrical gradient pulling that sodium in, whereas the electrical gradient is actually trying to hold on to the potassium, right? But there's a strong enough chemical gradient to allow some of it to flow out. But we got a much stronger push for sodium to get form of gradient. Okay, so acetylcholine receptors open and allow for sodium influx and potassium efflux. Okay, but much more of this one. Okay. So the end result of that is what's called depolarization. Means becoming less negative, right? We depolarize the membrane, it goes from that -70 up to something in the positive direction. Okay, if we get enough of that depolarization, meaning enough acetylcholine release, enough of those sodium rushing in, then that depolarization spreads over to this part of the membrane. And on this part of the membrane, we have these special ion channels, which again are voltage-gated ion channels. We'll talk more about those when we talk about action potentials, but that starts the action potential. Okay, so that's what's showing here. So this that depolarization leads to an action potential again, right? So we started with an action potential on the neuron. It took all those steps on the in between to get another action potential on the stuff. That's really the whole point of that. Another action potential there. Now we're back to where we started when we talked about excitation and contraction coupling, right? Action potential goes down the T2 right? So it's down right here. Okay, activates the subatomic reticulum to release calcium, right? And that's right back to where we want. Okay, so we do kind of do this backwards a little bit because we talked about monthly first, but then we'll take another step back next week and talk about how action potential happens. And we'll talk about kind of all the details of what happens in these little proteins to start an action potential. Okay, and then lastly, to terminate the stimulus, we got two options, the video talking about, right, acetylcholine esterase. How do we redo this? Yeah, ACHE, let's go with that, okay. Okay, so, yeah, ACHE, let's say acetylcholine esterase breaks down. Acetylcholine, okay, or it just diffuses out. Or diffuses out of the synapse. And goes away, so then without more acetylcholine release, then that active on muscle cell stops. And we stop the back to that muscle cell, stop the calcium, calcium goes back in the cytoplasmic reticulum. That's the component that moves, covers back up, moves the both the myosin, covers back up the myosin side, the muscle comes around. Okay, all right, so this quicker question, some of you have already answered it. If you haven't, go ahead and put your, put that answer in. All right, about 5 more of you or so, go ahead and put an answer in there. Okay. All right, so we're between B and D, almost exactly between B and D. Okay, turn to each other and convince your team of your answers. You can change it. You can change that. Fight for your answer. Talk to everybody. Okay, so change your answer if you want to. If you were convinced, then change your answer. Or if you're just stubborn, you're gonna keep it that way, then don't change it. Yeah, we have some malleable people out there, not too many though. We're about 2/3 on B now, about 1/3 on no, what was it? D, yeah. Okay, so we got 2/3 saying B, 1/3 saying D here, okay? This is a really, I really like what just happened right there, because this is a common misunderstanding, actually. What kind of transport does the sodium potassium pump do for it? We talked about, right? There's passive diffusion that could happen, right? That's just something moving with gradient. There's active transport, primary, secondary active. What type of movement does that sodium potassium pump do? Active, right? Active transport, okay. So when we talked about this right here, right? It's sodium rushing in, potassium rushing out. What type of transport is that? Or what type of movement is that? Does it take energy to move that sodium in the cell? No, that's passive diffusion. Right? Sodium going with its gradient, one concentration and electrical gradient. Okay, so those two things, the sodium potassium pump and sodium rushing into a cell to cause depolarization, separate those two things in your mind. Okay, but this is a common misunderstanding. Students commonly connect those two things. So I am glad that happened right there. So I can try to point this out. But the sodium potassium pump helps us maintain resting membrane potential, helps us return for that active potential, but it has nothing to do with causing an active potential. Okay, we talked about it right here, right? Here's sodium potassium pump. This is one of the things that helps us maintain active membrane potential, so it has nothing to do with causing an active potential. Okay, the active potential is caused by this passive sodium rushing into the cell. Okay, so the potassium pump will help recover from that, because it's going to pump that sodium back out, but that's actually the active potential. Okay, does that make sense? All right, good. Okay, well, let's talk about just a few, pathologies associated with neuromuscular junction. Okay, number one is an autoimmune disorder called myasthenia gravis. Okay, myasthenia gravis, is when our immune system, for some reason, starts recognizing our acetylcholine receptors as a foreign invader. And so attacking those acetylcholine receptors and destroys our neuromuscular junction. Okay, so this results in paralysis, weakness, things, symptoms like droopy eyelids, slurred speech, muscle atrophy, and we start losing our ability to contract muscle. And this is a pretty serious, autoimmune disorder, that is quite, quite familiar. Okay, so that's that one. Tetanus. So we talked about tetanus as far as the tetanus type, contraction, right? Nice smooth muscle contraction. But then there's also this kind of tetanus, which is an ontological tetanus. Okay, so this is due to a bacterial infection. The bacteria live in soil, dust, and manure. But then that can get into rusty metals too. Right, this one where we thought of tetanus when we cut by a rusty metal. We could end up with a tetanus infection. So this is something we have a vaccine for, right? The vaccine, if you've been vaccinated for tetanus, then you won't get this. Okay, but what it does is this bacteria releases an acetylcholine receptor agonist. And agonist is something that does the same thing that the acetylcholine does. Okay, so that means that it just binds to that acetylcholine receptor and causes activation of the muscle cells over and over and over again. So the muscles will start contracting uncontrollably, to the extent that they contract so hard that they can actually, your muscles can break their own bones when you have a tetanus infection. You have to rip your tendons right off your bones and actually break them. Okay. This one, there was a story that came out about this, about a family in Oregon a few years ago. And they were a family that didn't believe in getting any vaccines, and one of their kids got a tetanus infection. He was at the point in the hospital where he was on his deathbed, because if it gets into your diaphragm contracts, you can't keep breathing if your diaphragm is spasming. Went through this whole process, barely saved his life. And even after that, he still was vaccine. How this kind of stuff happens, but that was just their kind of relief, but he literally almost died from it. So no vaccine, we won't do it. But yeah, so that's 100% preventable with vaccine. So that's tetanus. Another one that's interesting is this chemical called Pure. Okay, Pure is a poison that is found on those little poisonous frogs in South America. Okay? So these little poisonous dart frogs, native people from South America have used that for years to put on the end of their darts, to literally, shoot an enemy and cause it to be paralyzed, right? So it's an acetylcholine receptor antagonist. Okay, so different than an agonist, right? An agonist does the same thing that acetylcholine does. This one does the opposite. It binds to it, but it gets in the way of acetylcholine actually binding to it, but it doesn't actually open up the ion channels like acetylcholine does. So you just can't have muscle contraction. Okay, so it can be fatal again based on same thing. If it gets to your diaphragm, can't contract the diaphragm, can't breathe. Right. One of the interesting parts about this is one of the drugs found in the cocktail used for lethal injection when somebody has a death penalty, enacted upon them. One of the things in that cocktail is a version of this chemical Pure. So that is Pure. Okay. All right. Well, we are done. Have a good lab this afternoon, and we'll see you guys on Friday for the test. I'll actually be here. All right, see ya. Bye, guys. Have a good one. See ya.
Good.
See you later. Yeah.
Just for that, is there a
Yeah.
lot of terms on the slides said like in photo,
Mm.
but then it's on there, so do we need to know that for the lot place? Like for the human, like how it's really important. Right. Okay, so like, okay.
Yes. Uh-huh. Mm, yes.